Well, hey, forensics friends. So, I got up here on the screen the DNA profiling activity sheet, and I thought we'd go through it, and you can see what you can get out of it. Just as a note, we're going to probably have some sort of final exam thing, and one of the things they'll ask is a little bit of DNA analysis, and that's what you'll need to be able to do. So, I don't know if you can read this, but follow along, and we'll see what we can do here. All right. So, let's see. The figures aren't showing here. Oh, fiddlesticks. But, all right. We're going to have the flanking sequence is the number of repeat units, and the number of repeat units here is showing as GAAP. And so what you look for is GAAP here in the sequence. So we got 1, 2, 3, 4, 5 GAAP sequences. So that would be for TPOXX, you'd have 5. And then the flanking regions are the regions on either side of it that are not part of the repeat, and so that's what you put down here. Let's see, we'll see if there's also there. You have then, this would be GATA. All right. And I say, crapola, this is not showing all the figures, but I'm going to keep on pushing through because I ain't going to jack with trying to download this thing. Hopefully you got it better than I sent it out. Oh, well. We have inheritance in that you're going to have one of the chromosomes from mom, one of the chromosomes from dad, and the chances are pretty good that they're going to have two separate repeat numbers, two different repeat numbers. And so that means that you're going to be heterozygous. And so if the parents are, like, say, dad has a repeat of 7 and then a repeat of 3, and mom has a repeat of 4 and a repeat of 3, it's possible for the daughter to get one of the 3 chromosomes from dad and one of the 3 repeat chromosomes from mom, and so she could be homozygous. This is sort of basic genetics here. All right, let's see. I wonder if I sent this to, this may not be what I sent you, because we didn't do student STRs. Here we go. So we got some profiles here. So these are the DNA profiles. Here's the suspect's DNA, and we've got a whole set of alleles here. And what you then do is fill in the profiles. Let's see here, and decide whether they're innocent or guilty. Okay, this was a complete and total mess. All right, but we're going to continue on, and holler to me if you aren't able to make any sense of this. The next thing is the allele frequencies. And allele frequencies represent the chance of, or the presence of this allele number out of 100, or actually out of 1, in the population. And the way you do the calculation is that if you're looking for the chance that they've got either this allele on this chromosome set or this allele on this chromosome set, that's 2 different possibilities, and there you begin to add the actual statistics numbers. Because if it's either this or that, you have the chances are going to be now greater, you're going to have one or the other. But now if it becomes you have to have this and that is you have to have a 3 on 5S818 and you have to have a 6 on CSF1PO, you're then going to have to multiply the numbers. And so when you multiply these small numbers, the numbers get even smaller and smaller. And so you start to have a really, really small chance that very quickly that it's not that individual's DNA is not a match. So let's see what happened here. All right. So then you can calculate up the probability of the different genotypes. And this is the way it works. Is you have P as the frequency of the first allele and Q is the frequency of the second allele. And this is a way of calculating up the statistics of the frequency or not. And you can calculate up the probabilities this way. All right, so what I'm going to be asking you on the, for the final is giving you something that'll look like this, where you've got a set of the CODIS spots, and you're going to have to identify the repeat sequence and then identify the number of repeats and then compare it to an unknown sample. So just in this case, you scan through and you see, all right, the repeat sequence is, let's see, AGAT, 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 and you just start counting up that number of repeats. AGAT. And here, the repeat sequence is looking around, GATA, 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 GATA. So you just have to scan through it and identify the repeat sequence, and from there you can count them up, and that's how you determine the allele number. All right, I'm gonna do some more discussion and looking at this, make sure that everybody knows what they're trying to do here, but hopefully we'll make the repeat sequences pretty obvious when we start looking at this. And I will post the video here, and I'll post a little discussion to see if you have any questions. That's all I have to say for now, and we'll check in when we meet up after Thanksgiving, and we'll do a little bit of review stuff. All right, guys, I'm out of here. Bye.